A very good morning, friends. Today we'll discuss about an important topic, that is purine nucleotide synthesis, or it is de novo synthesis of purine nucleotides. So first we'll see the sources of carbon and nitrogen in the purine rings. This is a purine structure. It is numbered in a clockwise direction. In this is the first, this is nitrogen, then this is N and this is N. It is numbered in a clockwise direction: 1, 2, 3, 4, 5, 6, 7, 8 and 9. There is double bond here. This is a purine structure. In this purine structure, this C4, C5 and N7, it is from the glycine. This N3 and N9, it is from the amide nitrogen of glutamine. This N from the aspartate, then C6 from the CO2, and this C2 and C8, it is from the tetrahydrofolate. So this is from N5 and 10 methynyl tetrahydrofolate, and this C2, it is from formyl tetrahydrofolate. So these are the sources of carbon and nitrogen in the purine rings. Coming to the de novo synthesis of purine nucleotide, there are three main sources or three main pathways for the synthesis of purines in our body. The first one is the de novo synthesis. Second one is the purine salvage pathway. And third one is the phosphorylation of purine nucleosides. Phosphorylation of purine nucleosides. Of this is a predominant pathway, but brain and RBC, they utilizes purine salvage pathway for synthesis of purines. So purines are dietary non-essential. They are synthesized from the amphibolic intermediates. So that is known as de novo synthesis. That is synthesis of purines from amphibolic intermediates. It is known as de novo synthesis of purines. There is MCQ from this sources of carbon and nitrogen. So remember this for the MCQ purpose. In the purine nucleotide synthesis, there is synthesis of IMP first. So from the pre-existing ribose 5-phosphate, so purines are build-ups on the pre-existing ribose 5-phosphate. There is synthesis of IMP first, and from this IMP you will get GMP and adenosine monophosphate, guanosine monophosphate, adenosine monophosphate. These monophosphates are converted into diphosphates, and these diphosphates are converted into triphosphates. And there is also in this diphosphate, there is conversion of ribonucleotide into a deoxyribonucleotide. So this is overall synthesis of purines, that is guanosine triphosphate and adenosine triphosphate. Now coming to the synthesis of IMP, there are total 11 steps in the synthesis of IMP, that is inosine monophosphate synthesis. Total 11 steps. Now remember this mnemonic. The steps 1, 3, 5, 6 and 8. These all steps required ATP and they are catalyzed by the synthetase. This is important to remember the enzyme, the intermediates. By this mnemonic 1, 3, 5, 6, 8, they require ATP and these all steps are catalyzed by the synthetase enzyme. Then step number 2 and 5, it requires glutamine. Third step required glycine. Fourth and 10th step required tetrahydrofolate. Okay. Then 7th step required CO2 and 8th it required aspartate. So in this way you can remember the steps in the IMP synthesis. So summarizing this, there are total 11 steps. So first, second, third. Now write down from here 4th, 5th, 6th, 7th, 8th, 9th, 10th and 11th. There are total 11 steps. So reaction 1, 3, 5, 6, 8, they require ATP. Now 2 and 5, this 2 and 5, it requires glutamine. 3 required glycine. 4th and 10th step required tetrahydrofolate. Nothing is required in the 9th step and nothing is required in the 11th step. So there is liberation of compounds. So fumarate will liberate from this 9th step and there is release of water molecule in the 11th step. In the 8th there is aspartate. 7th there is CO2. So by remembering this you can remember the entire pathway of IMP synthesis. Now coming to the IMP synthesis, the pre-existing molecule for purine nucleotide synthesis is ribose 5-phosphate. Now we will start the IMP synthesis. The first precursor molecule is ribose 5-phosphate, which is synthesized in the HMP shunt. Now this ribose 5-phosphate, in the first reaction there is utilization of ATP and this ribose 5-phosphate is converted into phosphoribosyl pyrophosphate. As there is ATP, it requires synthetase. So this is the PRPP synthetase. Now this PRPP, in the second step we required glutamine and this PRPP, by transfer of amido nitrogen to this PRPP, there is replacement of pyrophosphate and this PRPP is converted into 5-phosphoribosyl amine. So this is a amino acid, that's why there is amine, 5-phosphoribosyl amine. The important enzyme in this step is PRPP glutamyl amidotransferase. Now this 5-phosphoribosyl amine, there is glycine and this glycine converts this phosphoribosyl amine into glycinamide ribosyl 5-phosphate. So this is the first, second, third step. So third step requires ATP and it is synthetase. Now this glycinamide ribosyl 5-phosphate accepts the formyl group from N5 N10 methynyl tetrahydrofolate, and there is conversion of this glycinamide into formyl glycinamide ribosyl 5-phosphate. This is the fourth step. In the fifth step, this formyl glycinamide ribosyl 5-phosphate, there is glutamine and also ATP. So the enzyme is synthetase. So this formyl glycinamide ribosyl 5-phosphate, it is converted into formyl glycinamidine ribosyl 5-phosphate. So there is amino acid, so here glycinamidine ribosyl 5-phosphate. Now in the sixth step, there is ATP and ring closure occurs, and this compound, that is formyl glycinamidine ribosyl 5-phosphate, it is converted into amino imidazole ribosyl 5-phosphate due to the ring closure. This is the sixth step. Then in the seventh step, this amino imidazole ribosyl 5-phosphate, by the carboxylation reaction, it is converted into amino imidazole carboxylate ribosyl 5-phosphate. In the eighth reaction, this is carboxylase. This is biotin independent reaction. It does not require biotin. This amino imidazole carboxylate ribosyl 5-phosphate, there is addition of aspartate here. This is a amino acid. In the eighth reaction, it also requires ATP. This is converted into amino imidazole succinyl carboxamide ribosyl 5-phosphate in the eighth reaction. In the ninth reaction, nothing is added, so there is liberation of fumarate. This is with the help of enzyme adenyl succinate lyase. So there is lyase, adenyl succinate lyase. There is formation of amino imidazole carboxamide ribosyl 5-phosphate. In the 10th reaction, there is addition of tetrahydrofolate, that is formyl tetrahydrofolate, and this is converted into farm amino imidazole carboxamide ribosyl 5-phosphate. In the 11th reaction, there is release of water molecule, and this compound is converted into inosine monophosphate in the 11th step. So this is the cyclase reaction. This is transferase reaction, formyl transferase reaction. This is also transferase reaction. So this is a IMP synthesis. You can take a snapshot here. Now summarizing this, in the first, third, fifth, sixth and eighth step, there is addition of ATP molecule. So enzyme in these steps is synthetase. Then these two enzymes, PRPP synthetase and glutamyl amidinotransferase, they are important regulatory enzyme in this pathway. Then there is glutamine in the second step and fifth step, glycine in the third step, fourth and 10th step, there is tetrahydrofolate, CO2 in the 7th step and aspartate in the 8th step. So this is how you can remember the pathway of IMP synthesis. So there are total 11 steps. Nothing is added in the 9th and 11th step. So there is liberation of fumarate and water molecule. So this is in short, you can remember the IMP synthesis. Now from IMP, first we will discuss the regulation. So this is the regulatory step, committed step to this pathway, and it is under the feedback inhibition by the AMP, GMP, IMP. It is inhibited by these. Also, it is regulated by availability of PRPP, and this is also a regulatory enzyme in the purine nucleotide synthesis. So these two steps are important. There is increased activity of PRPP synthetase and glutamyl amidinotransferase in the primary gout. Now coming to the synthesis of GMP and AMP from the IMP molecule. This inosine monophosphate, it is converted into adenyl succinate, and this adenyl succinate is converted into AMP, that is adenosine monophosphate. This IMP, it is converted into xanthosine monophosphate, and it is converted into GMP, that is guanosine monophosphate. So this step, it requires aspartate. Also it requires GTP. There is liberation of fumarate. Then with the help of IMP dehydrogenase, this is NAD dependent reaction. So this is IMP dehydrogenase reaction, and with the help of glutamine and ATP, there is synthesis of GMP. So there is cross regulation. Synthesis of GMP requires ATP and synthesis of AMP requires the GTP. If one nucleotide is deficient, there is deficiency of also other nucleotide. So this is the importance of this cross regulation. Also these enzymes, that is adenyl succinate synthase and IMP dehydrogenase, they are under feedback control of these AMP and GMP. Now these monophosphates which are synthesized here, these monophosphates, so nucleotide monophosphates means AMP and GMP, they are converted into nucleotide diphosphates by the kinase, and these diphosphates are converted into nucleotide triphosphates means ATP and GTP by the diphosphate kinase. Here is the synthase or synthetase, and here there is GMP synthase. Summarizing the all, it is divided into IMP synthesis, then synthesis of monophosphate, and then synthesis of triphosphate. IMP synthesis is the 11 steps reaction. Then this IMP is converted into AMP and GMP, and by the kinase it is converted into nucleotide triphosphate. Now this nucleotide diphosphates or nucleoside diphosphate, it is converted into deoxy nucleoside diphosphate, by the nucleotide reductase. It is with the help of nucleotide reductase, and it requires thioredoxin and NADP. So this is all about the nucleotide synthesis, purine nucleotide synthesis. Another pathway for purine nucleotide synthesis is the purine salvage pathway, which requires the PRPP. Regulation mainly by the PRPP glutamyl amidotransferase, which is a committed step, and availability of the PRPP. These are the two main regulatory factors in the synthesis. So keep watching. Thank you.